Well, welcome to myDNA. For those that haven't been to myDNA before, I'm Alan Sheffield, one of the co-founders of myDNA, and we're really excited to have Sandosh with us from the NHS Scotland and the University of Glasgow to talk about a very exciting project that we've been working on over the last 4 years, and I think Sandosh has been working on it for 5 or 6 years to make this happen. He's down for a conference there's a pharmacology conference happening at the moment at the conference centre, and we thought it was too good an opportunity not to hold an industry event and just see some of the exciting things that we're doing with the NHS in Scotland, and particularly what Sandosh and the team have been able to deliver. I guess for those who don't know much about myDNA, I think most people in the room are enthusiastic about personalised medicine and preventative health, but me and my father, Professor Les Sheffield, started this business 15 years ago with a view of making pharmacogenomics more accessible. And when the University of Glasgow said that they were looking to roll pharmacogenomics to every member of the NHS, that really got us very excited, and we formed this partnership together. So it was very much aligned to our vision. And I guess we're just really excited to share the story and create some discussion. So Sandosh is going to share with you a lot about his experience over the last 4 or 5 years with pharmacogenomics, as well as the vision for the future of personalised medicine. And I think there's a lot that the UK and the Genomics England are doing that could apply to Genomics Australia here and to the industry. And I think everybody in this room is key to see personalised medicine progress and are advocates for what we do. And so thanks again for coming, and I might hand over to Sandosh.
Thank you, Alan, and it's great to be here. So I'm Sandosh Padmanabhan. I hold the Pontecorvo Chair of Pharmacogenomics at the University of Glasgow. I'm a clinical academic, so I mix research with clinical duties. The title of my talk is a question: what if every older adult had their pharmacogenomic profile on file? And I'm going to treat this as a practical question, not as a slogan. I'll work my way through 3 scenarios, and the answer turns out to be very, very interesting and a bit counterintuitive. So what is pharmacogenomics? Pharmacogenomics is, in one sentence, is simply that your genes change how your body handles medicines. How fast your body activates a drug, a prodrug, or how fast it clears the drug from the system, or how it converts to an active component. The defining feature about pharmacogenomics is that your DNA does not change throughout life. So your pharmacogenomic test result stays with you throughout life. One blood test taken at one point informs dozens or several dozens of prescription decisions across your lifetime. So this lifetime reusability is the promise. And the science of that is very clear, but we don't see this adopted into clinical practice. These are some examples, classical pharmacogenomic examples, and there are several others, and I'll come through those over the course of this talk. The first thing is the clinical problem. Why do we need pharmacogenomics? And I mentioned that it affects or it influences how your body responds to the drugs. And there's a huge problem with adverse drug reactions. Nearly 1 in 14 hospital admissions are caused in some form due to adverse drug reactions. And pharmacogenomics is one of the few tools available that can preempt adverse drug reactions and reduce morbidity and hospitalization. Routine testing is uncommon, and the reason for that is not because of lack of science or because science is missing. The reason for that is the health system does not have a settled answer to 2 questions. One is, who do we test and when do we test? If that's the case, what can we do to try and scale up pharmacogenomic use more broadly in the community? Let's go back to a few historical points about pharmacogenomics. It's important to know that pharmacogenomics is not something new. It's been well known for a long time. The term pharmacogenomics was coined in the 1950s. But even before that, you go back to 500 BC, Pythagoras mentioned a warned against eating fava beans, and that turned out to be G6PD deficiency. So knowledge of pharmacogenomics has been there for hundreds or even thousands of years. Even before the human genome sequence was published, we already had companion diagnostics for Herceptin, the HER2. It was before the human genome was published. We have guidelines starting in 2008 and 2009, guidelines informing or advising doctors on what to do if you encounter a pharmacogenetic variant. In 2023, we had the PREPARE trial in Europe covering 7 countries, one of the largest cluster randomized trial looking at the impact of panel pharmacogenomics on adverse drug reactions. And we have over 600 products with FDA labels indicating a pharmacogenomic interaction. So the point of this timeline is just to showcase that the science underlying pharmacogenomics is already there and quite mature. So if the question is no longer whether PGX works, the question is how to deploy it. Just look at the 2 important things which we have come to recently, which is data from the PREPARE trial. The PREPARE trial genotyped just over 6,000 patients, and they looked at a single drug pharmacogenetic test and a response to that. And they showed a 30% reduction in adverse drug reactions, which is quite a big impact of pharmacogenomic testing. They also, though it was not in their primary objective, they did a cost utility analysis, and they showed that it was cost savings in terms of, equivalent to about 12,000 euros per quality adjusted life year. Now, for adoption, the usual threshold is about 20,000. This shows cost utility. And they showed that it will be cost saving if the result is already on file for reuse. So it's not cost saving because the PREPARE trial was not designed for health economics. But even then they showed with reuse, you show cost savings. So in a sense, you have a large multinational trial showing both effectiveness, clinical effectiveness, and also evidence of cost effectiveness. Then if that's the case, then everything comes down to the practical skills, how to read a result and whom to give it to scale up pharmacogenomic adoption. And the important thing here is the pharmacogenomic result that comes to a doctor. It's not a verdict. And that's where we need to understand. It's an indication of about 5 actions. Whether to avoid a drug, so it's avoidable harm, or to choose an alternative drug, to adjust the dose, titrate the dose, or to monitor the patient differently. And most importantly, do not overact. Do not do anything. This is the decision support. This is the decision that goes to the doctor, and this is where some of the challenges are, because most of the physicians are not trained in pharmacogenomics. Getting them to understand requires quite effective decision support systems integrated into the healthcare systems. If this is how to act on the result, we've got good information. There are enough decision support systems available, and this can be, the science is known, so we can easily build these recommendation systems. The harder question is who should have the test now? And whom do we test? Do we test everybody? Do we test people when you're prescribed a drug, or do we wait for something bad to happen and then do the test? So we've got 2 types. One is reactive testing, wherein you do the test the moment you start the prescription. Preemptive testing is you have the test done preemptively when the patient is healthy, it's not on medicine, and you wait for a prescription to happen in the future, at some point in the future. And this one is the difficult question, because when you talk about preemptive pharmacogenomics, it means a huge upfront investment in getting genomics done. And pharmacogenomics is only useful when you're prescribed a drug. So you have to carry the variant that affects the drug, and you should be prescribed a drug. If you're not prescribed a drug, you may be very healthy all through your life and do not require drugs, in which case your PGX test is a wasted investment. So that's the problem. That's the unknown that's sort of blocking widespread scaling of pharmacogenomics, the upfront investment. And everybody, it's common sense that preemptive pharmacogenomics would be useful because everybody gets sick at some point and requires a drug. So how do we solve this problem? What I'm going to speak over the next half an hour or so is that we can use baseline polypharmacy, the baseline number of medicines, as a trigger to decide which testing group, is it reactive testing or preemptive testing, that we go forward. Before that, I'll give you an example. Meet Margaret, who is a 74-year-old lady with 6 long-term conditions, 9 repeat medications, started by 4 different prescribers over the last 10 years. Next week, she's expecting to be started on clopidogrel because she had a TIA, and because she's low in mood, she'll get an SSRI as well. So this is an example, a classical example of the patient we see quite often in a clinical practice. Now, those are 9 medicines. All of us have seen the prescription sheet. We've seen relatives with those prescriptions. But once you attach genes, pharmacogenes to those drugs, you see something different. You find that of all the drugs, 9 drugs, 6 medicines have pharmacogenes which are potentially influential in how she would respond to medicines. One panel could inform 6 of these medicines. And the real question is, does she need the result of the pharmacogenetic test before the prescription, or can we fetch it when the moment comes? Maybe can we wait until she... So that's the key question. So can we do reactive testing? Okay, or do we do preemptive? The important bit here is reactive is not wrong. It's early blind, meaning you do reactive testing when you prescribe a drug. But like Margaret, most of the patients are on multiple drugs. You get the result for one drug at that time of prescribing, but it's blind to all the other drugs. If you do reactive testing for clopidogrel, CYP2C19, it will be blind to the other drugs that she will be on. So she's getting SSRIs prescribed alongside, and that gets ignored. So reactive testing buys one answer at one moment for one drug. The question we need to ask is, for a patient like Margaret, is it worth having the answer for all the drugs? And would it be worth having the answer already on file so that expecting that she will be prescribed more drugs in the future, and that will shorten the time for her to get the right drug prescribed at the time of need. So what do I mean by this? Just look at her history. At age 60, she got prescribed atorvastatin, that's SLC01B1 pharmacogene. At 62, omeprazole, CYP2C19, 64, amitriptyline for pain, CYP2D6 and CYP2C19. At 66, codeine, then sertraline, clopidogrel. And 2 years later, she's going to get diagnosed with colorectal cancer, and we started on capecitabine. That's DPYD. So if she had preemptive testing, it's one panel being reused over the course of her trajectory. Her genome has not changed. The medication list changed 6 times. So if you have one result read once at 60, that would have informed every one of those decisions. Of course, the value of each answer depends on how actionable the pair is. But if you ignore that, it's the same result that's being reused. Even if it's not actionable, it is being used. A prescribing decision has been made or not made by awareness of that particular panel genotype. So again, if you revisit reactive and preemptive testing, we need a compromise. Test everyone or wait until something goes wrong. The compromise will be, as I mentioned, we need a trigger. We need a trigger to decide which one that we go for. So for this, we need evidence. And that's where we come to the Phoenix trial, which is a trial we are doing in collaboration with myDNA and Gene by Gene in Glasgow. And as Alan mentioned, there's a story behind it, but most importantly, you have a company in Melbourne funding a trial 17,000 miles away in Glasgow. But it's to answer a key question for pharmacogenomics. We want to evaluate clinical effectiveness of pharmacogenomics, but most importantly, cost effectiveness or a health economic case for pharmacogenomics, preemptive pharmacogenomics within the healthcare system. We started the trial on April 9th last year, 2025. The trial ends September 30th, 2026. So it's a very short trial. Our aim is to recruit up to 4,000 patients. We have randomized 2,470 as of yesterday. What we are doing is we are recruiting inpatients, so patients who get admitted to hospital. We have a data-driven strategy where the moment they get prescribed one of the 60 odd drugs that are part of the Phoenix trial, we get flagged, we go to the ward, consent them, and randomize them to immediate pharmacogenomic testing or delayed pharmacogenomic testing after 3 months. We follow them up for 3 months. We look at a lot of outcomes, from clinical outcomes to questionnaires, patient-reported outcomes. In Scotland, we have a unified health record system. Everybody has a unique ID. So even if they are admitted out with Glasgow in Scotland, we have access to their outcome data. So we'll have complete outcome data, we have complete prescribing data, and at the end of the trial, we will answer these questions about clinical effectiveness and cost effectiveness. So why do Phoenix? I mentioned the PREPARE trial already completed and showed adverse drug reaction, a 30% reduction in adverse drug reactions. Why do another trial? There are differences. Phoenix builds on and tries to answer more tougher questions than PREPARE. PREPARE used a 12-gene panel, about 38 drugs. We have about 13 genes for covering about 74 index drugs, but overall we have about 27 genes covered. The most important difference, PREPARE looked at one drug, preemptive testing for one drug. We have an index drug, but we also apply the pharmacogenomic results to all the patient's prescription. And the difference is we are looking at generating evidence for preemptive testing for a single drug, but we're also generating real-world evidence for reactive testing for the rest of the drugs the patient is on. So that's the difference. And it's embedded within the UK NHS system. Phoenix trial is on the web, all the details, the drugs that we are testing, all that's on the web. And these are the 74 medicines, and they convert to about 93 drug gene pairs because one gene can impact multiple drugs. And our primary objective, as I mentioned, we index the trial on one drug because that gives us a clean outcome. Then we have the real world evidence for all the other drugs. And we are measuring outcomes. So in contrast to PREPARE, the 30% reduction in adverse drug reactions was patient reported. It's not adjudicated hard clinical outcomes. Whereas in Phoenix, that's what we are doing. We do have patient reported outcomes, but our primary outcome is the hard clinical outcomes, mortality, hospital admissions. We also look at drug adherence. We pull their dispense prescribing and look at changes in their medicines. And so we have one clean endpoint, and we have the whole prescription for real world care. Because the trial is ongoing, whatever I'm presenting, the results I'm presenting are blinded. So I'm not going to give you any metrics by randomized arm. I'll give you the overall, because there's lots of interesting information there. This is the key bit. Who are the patients we've recruited? Where does the 2,000 odd patients come from? And what genes are though we are doing 13 genes, we see clustering of a few commonly implicated genes in these. Top recruiters, 20% are from cardiology wards, 13% from internal medicine, and 11% from general surgery. And we have a long tail of about 20 plus specialties, about 26 specialties, essentially the whole hospital. And this is important because it tells us that pharmacogenomics is not linked to a specialty. It's relevant everywhere any prescribing happens. And if you look at the description, this is not a tertiary care specialist hospital selection. This is a general case mix that you see in standard clinical practice in primary care or secondary care. So that's the population distribution. And who are the patients we've recruited? And again, this is overall. I'm not separating it by randomization arm. Average age 58. And we got equal number of males and females. It's predominantly white Scottish, slightly overweight. Of course, it's Glasgow, so 27 is the average. 26% of patients are obese. If you go to polypharmacy, and this is the interesting bit, more than 5 drugs, 78% of patients are more than 5 drugs, and 30% are more than 10 meds. But these are hospitalized patients, so it's skewed towards people who are multimorbid. 25% are current smokers, and strangely, only 9% have heavy alcohol intake. The other important bit to note is I skipped over this SIMD. SIMD is an area level deprivation, socioeconomic deprivation classification. And we have a bimodal distribution. And this is important because we have high numbers representing the most deprived and the most affluent groups. So if you look at SIMD, this is quintiles. Q1 is the most deprived, Q5 is the most affluent. And we got the whole spread. A third of the extremes we are recruiting. So we'll have good information on both. The smoking bit is important as an aside because it's a thing called phenoconversion. It's not just the genes. Sometimes it can be your environmental factors or your lifestyle habits that can change your phenotype. So that's the Phoenix population. Then we looked at, from our 2,000 patients, we compared to PREPARE. We looked at all the major genes, all the predicted pharmacogenetic phenotype, whether they are normal metabolizers, and we compared to PREPARE, and it's essentially 1. So our population is equivalent to PREPARE. That implies biological transportability, but clinical effects we need to do our study. But the most important bit is our genotype profile fits with PREPARE, and that's important for latter part of my talk. Now, what about actionability? Again, this is the whole population of Phoenix. We looked at not only what's happening with Phoenix now, but we also tried to look at what could happen in the future. Because as new drugs keep getting pharmacogenomic labels, the actionability or the number of genes relevant will keep increasing. So we divided it. The Phoenix is about 74 drugs. But if you look at all the drugs that currently have pharmacogenomic published guidelines, that's about 99 drugs. So if you look at the actionable drug-gene interactions per participant, if you just look at the Phoenix canonical number of drugs, it's 1.1. But if you look at the slightly larger 99 group of drugs, it goes up to 3.2. And then you go up to the universe of all the drugs in PharmGKB, because many of them will slowly filter down into guidelines. That goes up to 5.41. What this means is that in a real acute population, actionable pharmacogenomic results are the norm and not the exception. That's actionable somewhere. It's not a service plan, but it's actionable somewhere. And is there specific drug gene, actionable drug gene combinations that are sort of coming to the top? And what we see is about 4 gene core captures the majority of the targeted high-yield pathways. So CYP2C19, that's with clopidogrel and PPIs. Then you have SLC01B1 for statins, CYP2D6 for codeine, CYP2C9 for ibuprofen. And if you look at the major specialties, it's cardiology, gastro, and pain. They dominate the system. So that's Phoenix so far. We will complete the trial, and we'll hopefully have the results of the trial early next year. The other story is now, the same question is, okay, that's Phoenix. That's giving us controlled evidence, or it will produce controlled evidence through an interventional randomized blinded trial. What about the population level? When you want to implement pharmacogenomics, you're implementing at a population level. Whilst Phoenix will generate really important metrics, we wanted to look at the population and see what can we glean from the population on pharmacogenomic opportunity. Because in Scotland, like with most of the health systems, pharmacogenomics is not, they're all PGX naive. So Pharmacogenomics not used. What we want to know is what's the potential opportunity for pharmacogenomics if we implement pharmacogenomics, and what would be how do we implement pharmacogenomics so that everybody benefits. What we have here is, because of our healthcare data system, we have a real world lens on exposure, equity, and complexity at a population level. We looked at data on everybody who were 51 years or above in Glasgow on January 1st, 2012. That's about 471,000 patients, all 51 years old. About 65% of them received at least one PGX implicated drug in 1 year. We just looked at 2015, and at least 60%. If you scale that to the Scottish population of over 50 years old, that's about 1 million patients. So 1 million patient gets exposed to at least one pharmacogenetic drug within 12 months. And that brings the concept that it's so pervasive that when you talk about pharmacogenetics, it has to be infrastructure, not as a special test. So these are the numbers. So we looked at data in 3 snapshots, 2016, 2019, 2021. This is a closed cohort, meaning I got everybody over the age of 51 in 2012 and got all their data, healthcare data, dispensing data, everything until 2022. So that's 10 years of data. And we got prescribing, hospital admissions, death records, deprivation, drug gene mapping. We got all that and did a lot of analysis. And I'm going to quickly go through a few results. And I won't go into the details because it's beyond the scope of this talk, but it'll give you some headline insights. The first result we got is entry into pharmacogenomic prescribing. Is that when does a patient get their first, or who are the patients who get their first pharmacogenomic implicated drug? We looked at multimorbidity, we looked at deprivation, and we looked at polypharmacy. And the best results are with polypharmacy. So if you look at polypharmacy, what we find is polypharmacy, the baseline number of drugs at any point predicts entry into pharmacogenomic prescribing. So if you're on 5 baseline drugs, you have a 35% higher chance of starting a new PGX drug. Meaning you're on 5 drugs, but none of those are pharmacogenomic drugs. So you're on polypharmacy with non-pharmacogenomic drugs. The higher the number of drugs you're on, you have a 35% plus chance of starting a drug in that year. What does that mean? That means medicine count flags the patient about to cross into a territory where a pharmacogenomic test would pay off. In contrast, so as I mentioned, with entry into a new pharmacogenomic drug, the number of baseline drugs predict. What about intensification? If you're already on one pharmacogenetic drug, do you cluster? Do you start piling up more pharmacogenomic drugs? And that's 0. So polypharmacy, the number of drugs that you're on, can predict entry into new pharmacogenomic prescribing, but it does not predict. There's no relation between that and piling up multiple drugs within a year. So that's an interesting insight, which is new. What about the cumulative value? We all say that the drug panel can be used and reused over time. What's the value? So we looked at, again, 3 groups of drugs. We looked at how many drug-gene interactions can a panel of tests inform over 5 years. So we looked at the PREPARE panel, the 12-gene panel. We looked at the sort of slightly expanded Phoenix panel, which is the guideline tier. And we looked at the total PGX, the universe of pharmacogenomic drugs. And what's the number of drug-gene interactions impacted over 5 years? And that's about 78. 78 drug-gene interactions impacted for the current guideline group of drugs. If you look at the whole pharm GKB, the full universe of drugs, is 145. We also did some sensitivity analysis. So you could say that, but this is a closed cohort. So over 10 years, people are dying. So the number of people comes down. So when we did the survivor cohort, everybody who survived the whole 10 years, the number is still the same. It's about 69 drug-gene interactions are informed over a 5-year period. So you can see the compounding happening with pharmacogenomic tests. panel tests. What about value? Now, we look at two metrics. One is yield, and that's the left side graph. And what we show is, so the important thing here is, this is where I'm bringing the PREPARE alignment with our Phoenix trial. PREPARE published health economics. We transport the PREPARE metrics into our population, 470,000 patient metrics, to build this. That's more accurate than us modeling scenarios. This is still scenario-based implementation, but it gives you some idea. So what we have is that yield rises with baseline polypharmacy. So the more drugs you are at baseline, the yield, the number of DGI per eligible per patient increases. So from a person on one baseline drug to a person on 5 baseline drug is from 24 to 58 DGI. So that's the rise in DGIs. What about value? And the cost per quali declines from 13,000 to 5,000. So if you were to target pharmacogenomics based on polypharmacy levels, you get more value if you first target, everybody needs the pharmacogenetic, but if you first target the people with multiple drugs and then go backwards. We looked at multimorbidity, we looked at deprivation to see whether can we use multimorbidity as a trigger, or could we use deprivation as a trigger, because we know deprived individuals have more morbidity and they're on more drugs. But all the scenarios showed that polypharmacy is the best metric. Multimorbidity is good, but polypharmacy, that metric is already available in the health records readily. Multimorbidity has to be calculated, and there are a lot of errors, missed diagnosis, which can impact that. Whereas medicine is just a prescription and it's a count. It's readily available at no extra cost. So we looked at multimorbidity and deprivation, and what we show is polypharmacy, and this is where this modeling showed polypharmacy captures 67.9% of eligible patients, whereas multimorbidity captured 65%. This is modeling assuming that you won't be testing everybody. So if you test about 40% of the cohort, so from about 35% to about 70% of the cohort, if you test that range of testing, at every point, polypharmacy is more favorable. Deprivation is interesting. It's a poor trigger, but what we show is this is neutral. If you trigger pharmacogenetic testing using polypharmacy, it captures inequity. You don't amplify inequities or disparities by this method. And does it pay? If you take the PREPARE economics and transport to Glasgow 400K patients, there are about 138,000 high medication eligible patients, more than 5 medicines in one health board. The PREPARE health metrics, when you transport it, we are saving about 245 to 400 quality adjusted life years, and that's equivalent to about 12 to 15 million in avoided downstream costs. This is illustrative because we will get more actual data once Phoenix publishes, but we don't expect it to deviate much from PREPARE. So how do we scale up? How do we implement pharmacogenetics? So this is based on all the analysis that we have done and I've presented so far. If you want to implement pharmacogenetics, the first thing is a basic thing. Is it reactive or preemptive? And from the data I said, the trigger would be the first prescription of a pharmacogenetic drug would be the trigger for reactive testing. So you're getting people into the pharmacogenomics. You're not doing a blind all-comers pharmacogenetic testing straight away. So patient gets their first do pharmacogenetic testing. Separately, patients who are on 5+ drugs, genotype them, all of them, because that's preemptive for them. So that's the hybrid model, which means ultimately you will end up with everybody having a pharmacogenetic test on 5, or every older adult having a pharmacogenetic, but you're staging it in a way that you maximize impact and everybody benefits from PGX testing. In the UK, we've been thinking about how can Scotland or even whole of UK use this information to inform their implementation. So what we have in the UK currently is a very small number of reactive pharmacogenetics, so DPYD, mtRNR for aminoglycoside in neonates, and clopidogrel for stroke coming online soon. That's it, a small handful of drug genes in a reactive form, because the healthcare lab, our lab capacity is limited. It has to cover all the possible genomic testing. So that's what we have. But what we don't want is to break the system by suddenly saying everybody needs pharmacogenetic testing. So what you do is you do a staged, stratified reactive in stage 1, which is what I mentioned, polypharmacy as a trigger. If somebody started on a new PGX test, do reactive testing, and for greater than 5 drugs. Then you do targeted preemptive in years 3 to 5, and ultimately it will naturally progress. And the reason for that is straightforward. You want to use existing infrastructure. So a stratified reactive is good because you just have to slightly scale up what we already have for reactive pharmacogenetics. The people we need, reactive testing are basically specialist based, and there's no health economics. It's only cost avoidance for reactive testing. When you scale up, you really need to show health economic, you need to show cost savings. So that's why the stratified reactive means you don't need a lot of infrastructure changes. You need some champions, and it's cross-sector. Then as you scale up, then it becomes you need your infrastructure to become more automated. Then you start more training, education, everything. And then ultimately it moves on to a learning health system and with universal testing. So in summary, what I'm suggesting is reactive PGX is a stepping stone. And each stage delivers benefit while you build capability for the next. The question for health systems is that it's no longer does PGX work, it's whether we are willing to treat it as infrastructure. And for the Phoenix trial, huge amount of work with a lot of people. Stephanie is here, who is driving all the recruitment and the whole operation of the Phoenix trial. And huge support from myDNA, Les, Alan, Sam, and Rachel, and everybody else. Thank you.